right, guys, let's get back into decoding your future. This is going to be part 2, so let's just go ahead and jump on into it. Now, just a few questions. What organelle is responsible for making proteins? Do you remember? Ribosomes. Good. Where are ribosomes found in the cell? In the cytoplasm and the rough endoplasmic reticulum. Very nice job. See, I knew you guys were smart and paying attention. If DNA is found in the nucleus and has the genetic code, then how does it reach the ribosomes in the cytoplasm or on the rough endoplasmic reticulum to make proteins? RNA. Very nice. Now, what is a protein? So a molecule is formed by the linking of amino acids, which we already know because we talked about in our biomolecules unit. Now, there are 20 different amino acids. The roles of proteins can be to make enzymes, they offer structural support, they offer antibodies to you, and they make hormones. Now, let's get into gene expression. The instructions for building a protein are found in a gene and are rewritten to a molecule of RNA during transcription. The RNA is then deciphered during translation. So just remember, you go from DNA to transcription to RNA to translation, which makes proteins. Now, what is RNA? This is another type of nucleic acid, so its building block is also going to be a nucleotide. But what makes it different is the sugar and one of the nitrogenous bases. Each RNA nucleotide contains a phosphate. Their sugar, instead of deoxyribose, is just ribose. And they have 4 nitrogenous bases, but notice something different. They have adenine, uracil, guanine, and cytosine. Did you catch the difference? Well, if you didn't, let's go back. Uracil, that wasn't something that was in our DNA because we had thymine. So instead of our A and our T coming together, our A is now going to bind with our U when it comes to RNA because we have no thymine. And RNA is a single-stranded molecule instead of a double-stranded molecule. So here are your 3 main differences from RNA and DNA. RNA is single-stranded while DNA is double-stranded, so the D in DNA double. Keep that in mind. Now, RNA sugar is ribose. That's what the R in RNA stands for, while DNA sugar is deoxyribose. And finally, RNA has the nitrogenous base uracil, while DNA has thymine. Those are your 3 main differences between RNA and DNA. Now, there are different types of RNA, and here's the first one. You have mRNA, which stands for messenger RNA, which functions to carry messages from the DNA to the ribosomes. So it's the little male guy that's running over to be like, hey, this is what you need to make. Now we have tRNA. This is our transfer RNA that functions by carrying amino acids from the cytoplasm to the ribosomes, where it adds the amino acids to the protein chain. So it's transferring from one thing to another. Next, we have rRNA, which is our ribosomal RNA. So its function is to form ribosomes, and this just happens to be the site of protein synthesis. Now, protein synthesis happens in two different stages: transcription and translation. In the first stage, which is transcription, to transcribe basically just means to copy something down, just like somebody in a courtroom taking notes, or the person who follows the doctor around writing everything down they say. So DNA is copied to an RNA molecule called mRNA, and this all occurs in the nucleus of the cell. Now, translation means to change from one language to another, which I'm sure you guys know that because a lot of us are translating stuff every day. Now, the information is translated from the language of RNA nucleotides to the language of proteins, which are going to be our amino acids, because amino acids build our proteins. Now, this is going to occur in the cytoplasm of the cell at the ribosomes. So let's break transcription down. So step one is that RNA polymerase binds to DNA at a location called the promoter. So here is a promoter site on DNA, and this is your RNA polymerase. So that polymerase is going to bind to the promoter to start transcription. So the RNA polymerase will work to unwind and separate the two DNA strands. So as you can see, we are separating our DNA strand here by using RNA polymerase instead of DNA. Now, step 3 of transcription. RNA polymerase adds and then links complementary RNA and nucleotides to make mRNA. So this begins at a start sequence and ends at a stop sequence. Now, our base pairing rules from RNA to DNA are going to be A pairs with U, T pairs with A, G pairs with C, and C pairs with G. That is how that is going to work for DNA to RNA. So we have our DNA double helix reforming here as our RNA is coming in. Now, step 4. Before the mRNA can leave the nucleus, it is modified. Now, mRNA has coding regions called exons. mRNA also has a non-coding region, called introns. Now, the introns are cut out of mRNA, and the exons are spliced or glued together. Spliced is just a fancy word for saying we're gluing stuff together. Now, let's take a closer look here, because you know me, I am all about pictures. Pictures help us so much. So here, you have your chromosomal DNA, and you have exon 1, exon 2, and exon 3. And we have these introns that are inserting within them, because this is one gene as a whole. Now, through transcription and RNA synthesis, we're going to see our 3 exons and our introns as well, but with our nuclear RNA. Now, in the process of RNA splicing, we are going to cut out these introns to glue together our exons to make the messenger RNA. And that's how RNA synthesis and processing occurs. Step 5. RNA will then leave the nucleus and it goes to the cytoplasm to find a ribosome for translation, which translation just happens to be step 2 of protein synthesis. Now, let's do a quick review of transcription from DNA to mRNA. Our DNA nitrogenous bases are A, T, C, and G, which we already know from our last PowerPoint. But our RNA nitrogenous bases are going to be A, U, C, and G. So remember, we're replacing that T with our U in RNA. So our base pairing rules DNA to mRNA. So G to C, A to U, T to A. So an example of this would be, here is a DNA sample. You have all your regular base pairs for DNA, but here's how it would be transcribed into RNA. So instead of this turning into a T, it's a U. Let's talk about codons real quick before we get into translation. Now, a codon makes up mRNA. So they are made of 3 nucleotides on the mRNA molecule. So each codon codes for a specific amino acid. And it also can have your stop or start signal, which I'll be able to explain it better when we have a molecule to look at. So our codon is going to be made up of our base pairs, and it's going to code for our amino acid, and that's how we build the protein. So here's a better picture of it. So this is one codon. Each codon has 3 base pairs in it. Your 3 base pairs makes one codon, which makes one amino acid. Now, there are going to be 20 different amino acids, but there are 64 different codons in the genetic code. 61 codons will code for amino acids. 1 will equal a start codon. Now, 3 codons signal the stop of protein synthesis. The order of the nitrogenous bases on the DNA strand will determine the order of amino acids in a protein. The genetic code is the same for every organism, which means that the codons represent the same amino acids in all organisms. So here is one way that we can read our codon chart for mRNA. So let's say you have the codon UUU, for example. Because as you can see, we have so many different variations. So the way that you would read this is this column is your first base pair. So our first base pair was U. So we would be working with this row. Now, up here is where we find our second base pair. So we know that if it's UU, we are only going to be looking at these codons. Now, finally, we have our third base pair, which is over here, which I said was UUU. So we know that we need to go all the way across the top. UUU codes for this. Now, whenever we're writing these amino acids down, we don't always write the full amino acid. A lot of times we'll just write the first 3 or 4 letters. Most people know what you're talking about. But that is how you read the square version of the codon chart. Now, this is a circle version of it. I like this one a lot more. It makes a lot more sense to me. So you read it the same way. Let's say we're doing UUU again. So we find our U, we find our U again, and so you just kind of follow it out of your circle till you get to the exterior. You may use whatever chart you would like. I just like this one. I feel like this one is easier to read and to understand. Let's talk about tRNA, which is just a reminder, your transfer RNA. So tRNA reads the mRNA and brings the correct amino acid. So at one end of the tRNA is the anticodon, which is complementary to the mRNA codon. So the rules of this are going to be A to U and G to C. Now at the other end of the tRNA is the amino acid. So you see we got our anticodon down here and we got our amino acid up here. So let's talk about translation steps. mRNA attaches to the ribosome, which is made of rRNA or ribosomal RNA. So the mRNA starts at AUG. This is our start codon. So if you see the AUG, you know that is where your translation begins. So mRNA ends at UAA, UGA, or UAG. This is your stop codon. So you know that whenever you come to this, you cease production immediately, even though there are complementary base pairs to keep making codons. As soon as you see stop, you stop. Now, the first tRNA molecule reads the mRNA by using an anticodon, and it also brings the first matching amino acid. Now, the ribosome moves down the mRNA so another tRNA can translate the mRNA into amino acids. So the tRNA keeps bringing amino acids until the stop codon is reached. So the amino acids are then going to be connected by peptide bonds. So let's think about the way that this works. So the tRNA is bringing amino acids because the mRNA is telling it to. So I'm sure you guys know how some of our grocery stores can do online pickup. So imagine you are the messenger RNA. You're saying, okay, I need milk, bread, and eggs. And the tRNA is your shopper that's getting your order together in the grocery store. It's like, okay, my mRNA told me I need to get this, this, and this, and this. So I'm going to go get it, and I'm going to bring it back to you because this is what you need to complete your order or to complete your amino acid chain. All right, so we got our mRNA coming from the nucleus, and down here we've got our ribosome. So you see that our tRNA, this is where we're starting. See? AUG, that is our start codon. Now here is going to be our anticodon, which our tRNA will read to get our amino acid, which is, we don't have an amino acid for AUG, it's just a start codon. So we're going to keep building on. So we're going to convert our base pairs to RNA. Our tRNA is going to go get our amino acid. And it's going to continue down the line. Our tRNA is going to continue getting the stuff for us. So if you notice, we came to a point that we had to stop. And this is what your protein would look like. You'd have your start codon. You'd have the amino acids that built it up, and then your stop codon. And this is your protein. So the way that we read codons is we read it 3 base pairs at a time. So the first 3 here, which is GCU, that's going to be codon 1. ACG, codon 2, and so on and so forth. And every set of 3 is going to continue making codons. So then, these codons is what we're going to use to read for our genetic code. So let's read the first one, for example. GCU. So we'll have a G. Here's our first pair. So a G. Our second base pair is C. So we're going to be down here. And U. So you're going to follow that across for the U. And that'll give us alanine as our amino acid to build our protein. So here's a better representation to show you how we code for that first codon and using our code reader. Now, let's look at codon 6. So our first one is A, so we're going to be in that column with our A for our first base. Our second base is going to be G, so we're going to be in the far right column. And our last base is C. So we know our amino acid is going to be serine. Now, just to review from translation from mRNA to amino acids and protein. So our RNA nitrogen bases are going to be AUCG. Our base pairing rules from mRNA to tRNA anticodon are G to C, A to U. At the other end of the tRNA is the amino acid. So we use our codon charts to determine the amino acid based off of mRNA, not the tRNA. So an example of this would be, we have our mRNA strand here. So we then have to transcribe it to tRNA, and that is what we use our codon chart for. So we have our start codon and our 2 amino acids. All right. And that is going to be the end of our lecture on transcription and translation and RNA. I hope you guys